Okay, so today we're going to talk about mutations. Your objective is to be able to explain how mutations in DNA that result in interactions with the environment lead to changes in function and phenotype. Phenotype is a physical characteristic that you actually see. For example, you can actually see freckles, uh, brown hair, brown eyes. Those are characteristics that you can physically see. A long tail on a dog, a short tail on a dog. All of those are phenotype. So what are mutations? Mutations are changes in the nucleotide sequence of DNA. What are the three parts to a nucleotide? You should have said phosphate, sugar, and base. A mutation can occur in any cell of the body. If it occurs in your somatic cells, those are your body cells, you don't pass that on to your offspring. And if it occurs in your gametes, that's the egg and sperm, it can be passed to your offspring. So are mutations always bad? Not necessarily. They may look strange, but that doesn't mean it's going to harm you or kill you. So it's not necessarily bad. Mutations happen regularly. Almost all mutations are neutral. That means it doesn't hurt you and it doesn't do anything good for you either. Chemicals and ultraviolet radiation can cause a mutation, and many mutations are repaired by enzymes. Some types of skin cancers and leukemia can result from somatic mutations. Somatic, again, are cells of the body. Some mutations may improve an organism's ability to survive. It may be beneficial for an organism. So what are the types of mutations? We have two main types of mutations. We have chromosomal mutations. Chromosomal mutations happen during meiosis. It changes the structure of a chromosome. Examples would include Down syndrome, Klinefelter syndrome, Turner syndrome. And gene mutations, they happen during DNA replication. That's a change to the original DNA nucleotide sequence. And examples of gene mutations would be cystic fibrosis, dwarfism, and sickle cell anemia. So a chromosomal mutation involves changing the actual structure of a chromosome. You're either going to lose or gain a part of a chromosome. We have 5 types of chromosomal mutations. We have a deletion, inversion, translocation, non-disjunction, and duplication. A deletion is simply that. It's due to a breakage. A piece of that chromosome is lost, but the chromosome reattaches itself together, but that piece of DNA, that piece of gene, is actually deleted. An inversion involves the chromosome segment breaking off, flipping around, and reattaching itself. Flips around, it reattaches itself. Duplication, exactly what it sounds like. The sequence is repeated. Translocation involves 2 chromosomes that are not homologous. Homologous means it's for the same trait. Like you get one chromosome from mom, you get half your chromosomes from mom, you get half your chromosomes for dad. So those chromosomes are for the same trait, like for hair color or eye color. If they're not homologous, one may be for hair color, the other may be for height. That means they are not for the same trait. Part of one chromosome is transferred to another chromosome. And you can see over here, before translocation, A and B, one is light, one is dark, and we're simplifying this, of course. But after translocation, A has a piece of B's on it, and B has a piece of white. Non-disjunction is the failure of chromosomes to separate during meiosis. It causes the gametes to have either too many or too few chromosomes. Disorders because of non-disjunction include Down syndrome. Down syndrome, you have 3 chromosomes on chromosome number 21. Turner syndrome are females, and instead of having 2 X chromosomes, they only have 1 X chromosome. Klinefelter syndrome are males, and instead of having just an X and a Y as a male, they have 2 X's and a Y. This is non-disjunction and displayed on meiosis. This is the starting parent cell. The result is 4 haploid daughter cells. And on this one, each daughter cell should have 2 different chromosomes. But you can see here, this has 3, this has 3, and these 2 have 1 each. So they didn't separate properly. So this is an animation of chromosomal mutations. A gene mutation is a change in the nucleotide sequence of a gene. It can involve just one single nucleotide. The mutation can be due to copying errors, chemicals, viruses, etc. Gene mutations include point mutations, substitutions, insertions, deletions, and frame shift mutations. A point mutation is a change in one single nucleotide. It includes the deletion. It can be because of a deletion, an insertion, or a substitution of one nucleotide. For example, ATT-ACC, we can if it's substituted the T for the A, that's a point mutation. An example of a point mutation would be sickle cell disease. It occurs because of one single nucleotide substitution. It occurs in the hemoglobin of the gene. As you know, the hemoglobin delivers oxygen to your cells, and if it can't deliver oxygen, right, it can't deliver it because the red blood cell is shaped like a sickle or like a quarter moon. So it doesn't give all of your cells that needed oxygen. A frame shift mutation is inserting or deleting one or more nucleotides. All codons that come after the change is affected. It changes the reading frame like changing the sequence of a sentence. And because it can't build the protein correctly, the protein may not be built. So for example, the ribosome reads the messenger RNA 3 bases at a time, right? So ATT-ACC, if one of the bases were deleted, that second T in the first codon, it would read it ATA, which is of course a different amino acid. If this is an insertion, right, we inserted a C in that first codon, so it would read as ACT instead of ATT, and that also would be a different codon. Another way to look at it as changing the reading frame, if we had the sentence, the fat cat ate the wee rat, right? If we insert an extra A in cat, we can no longer read that sentence. You can't read it after the fat. That's the same thing that happens in building those proteins. If the ribosome can't read what amino acids it's supposed to be building, it can't build the protein correctly. So it changes the reading frame. It changes how the protein is built. Here's another example of an amino acid change. We have our DNA molecule right here. We've already transcribed it into messenger RNA, and these are the amino acids. If we were to take out the G, or a mutation occurred and the G was deleted in this DNA sequence, instead of GLU, we would get LYS, which would lead to a different protein being built. Here's another gene mutation animation. And just for your information, this is called a karyotype. A karyotype is a picture of chromosomes taken during metaphase. It's in size sequence. This would be chromosome number 1. This would be chromosome number 22. And the 23rd pair gives you the sex of the individual. So this would be a normal male. This would be a normal female. She has 2 X chromosomes. This would be a male with Down syndrome. Trisomy means that it's on that chromosome number, meaning it's 3 chromosomes. And this particular one, 3 chromosomes on number 21. You should only have 2, one from each parent. So this male has he has 1 X chromosome and 1 Y chromosome. So this male has Down syndrome. This female has Down syndrome, 3 chromosomes on chromosome number 21. And this man has Klinefelters. He has 2 X chromosomes and 1 Y. And this female has Turner syndrome. She only has 1 X chromosome.